Hi, everyone. It's my great pleasure to introduce Meghan Driscoll for our Center for Health AI seminar series. Dr. Driscoll is currently an instructor at the University of Texas Southwestern Medical Center, where she studies dynamic cell morphology, computer vision, and 3D microscopy analysis. She is currently supported by a K99 and was previously awarded an F32, so two very prestigious awards to have won. Before she was an instructor, she was a postdoc also at UT Southwestern, but she's also speaking to us today coming from a background in physics. And it's always fun when physicists focus on biological problems. She received her PhD in physics from the University of Maryland, College Park, as well as a bachelor's in physics from Harvey Mudd College. So I won't try to spoil things that she's going to talk about, but I am familiar with her work, and she's recently written a series of rather compelling articles, some that are focused on biology and others that kind of lean towards the software. So I think I won't spoil any of the fun that she's going to talk about, but I did want to highlight one particular article, which is a perspective piece that she put out almost a year ago titled Data Science and Cell Imaging. And so I'm not sure if you're going to talk about this at all in your talk, but I found it a really amazing article, and it's required reading in my lab now. So with that, I'll let you take it away.
Well, thank you for that intro, and I'm glad that people are reading my articles. There's always this fear that you're sending things out there and no one is reading it. So I'm glad to hear that people are reading it. Right, so I'm excited to be here today. I'm going to talk about the functions of cell morphology, and cells have very varied and diverse forms, right? So this is an immune cell on the left. It has sheet-like protrusions called lamellipodia, right? This is a melanoma cell in the middle that has these hemispherical protrusions called blebs, right? And this is a model for lung cancer on the right that has those spiky protrusions called philopodia. Right? And these are not unusual cell morphologies. When we begin to look closely at cells, we see that they have crazy and intricate morphologies, and these morphologies have function, right? So the branch morphology of neurons allows cells to connect to one another, enabling thought, right? Cilia in the lungs help the lungs to function, and the dynamics of form also have function. So here, these are cancer cells metastasizing in a mouse, right? The cells are, the cytoplasm of the cells are labeled in red, right? And the nuclei are labeled in green. And we often understand migration as a problem of change in dynamic cell form. And so cell form and shape is really regulated at the local micron scale, right? So signaling within the cell controls cytoskeletal dynamics, which is the stiff part at the cell edge that changes cell shape. And that in turn drives cell form dynamics, right? So although we're used to thinking about the cells as a sort of bag of signaling, they have very localized signaling and localized cytoskeletal dynamics that in part allow them to move, okay? These are two actin cytoskeletal regulators in a breast cancer cell. Although biology as a whole really has many tools now to interrogate the central dogma scale problems, right? Even if we were to completely understand how DNA relates to RNA and proteins, that wouldn't be enough to understand cell form, right? And that's in part because cell form is emergent. We need to understand how physical forces and the organization of the cell comes together to create cell form, but also because cell form is hard to perturb. We can perturb a single protein, we can change an amino acid, but we can also change cell form, but it's hard to interpret what that means. Yeah, I can chop off a philopodia, but probably that will not tell us very much, right? And another problem is that only very recently have we been able to actually measure cell form at the relevant scales. So it's surprisingly hard to actually, with a microscope, image at that scale, right? So when I started in grad school, I intended to do more standard physics, but I really fell in love with cell shape. I studied amoeba, no actual amoeba. And even though amoeba are sort of the quintessential, have quintessentially crazy three-dimensional shapes, we didn't have the microscopy at that time, really anywhere, to understand, to image cells in 3D, okay? So instead, I imaged cells on strange three-dimensional environments like cliffs. I tried to drive them off the edge of a cliff. But as a postdoc, I really wanted to be able to study cells in 3D because cells are fundamentally 3D objects, right? So we want to be able to image them in 3D. And also cells adapt their form to their environment. So if we take a melanoma cell and put it on a hard surface, it will splay out on that surface, right? It will stretch itself out really almost as thinly as it can, right? So you see that on the left. Whereas if we take a cell and put it in 3D collagen, in a soft collagen matrix, or in the brain of a zebrafish, those same melanoma cells will ball up and wrinkle their morphology, right? So they have wildly different morphologies depending on their environment. Right? And recently, thanks to advances in microscopy, we can actually now image the cell with enough spatial resolution to see signaling in subcellular dynamics, right? So we have genetic engineering that allows us to engineer in fluorescent proteins that light up elements of the cell that we're interested in. And we have high resolution light sheet microscopy that can image in 3D at speeds fast enough to capture cell movements, okay? So this is, I think, a great time to be in the field because we can only now actually tell what cells are doing. And so as a postdoc, we sought to study cells in 3D, and the first thing we did, which was really a group effort, was develop a microscope to study cells at high resolution and in soft 3D environments. So this microscope has nothing hard in it. There's no cover slip at all, right? Just a hunk of soft material of various types, right? And then usually it has isotropic resolution. So many microscopy modalities are worse in Z, right, than in XY. Here we have roughly 300 nanometer resolution in all directions, okay? All right, so then Eric Welf and I now many years ago decided to study how melanoma cells migrate in these soft matrices. So some human cells, like amoeba, are considered amoeboid, right? And melanoma cells can adopt this rounded amoeboid form, though they also sometimes adopt a more mesenchymal branched form, right? But we found with primary cells that were grown in mouse xenografts that the more metastatic the original tumor, the more amoeboid the cells were. And other people have found this as well in various contexts. So we know that an amoeboid form is associated with metastasis for melanoma cells. Okay. So we wanted to know how melanoma cells migrate, right? If you just put melanoma cells on a cover slip, they tend to adopt a mesenchymal form, right? So it strangely, as a field, we still don't really know how melanoma cells migrate in 3D. Although there was a very well-known model at the time of amoeboid migration. That model is that cells deform their bodies in order to sort of snake through the very crowded environments found in vivo, and that they find the path of least resistance in order to push their way through, okay? But this model was really largely, I think, informed by immune cell migration. So there's a really great set of papers from Michael Sick's lab, where they studied how dendritic cells move through 3D environments. And dendritic cells are considered professional migrators, right? They're sentinel cells that patrol your body and look for pathogens. And once they find a pathogen, they try and jumpstart adaptive immunity, right? By some cases migrating to somewhere like a lymph node, right? So these cells are very good at migrating. So I went to Michael's 6 lab and took these cells. I normally focus on image analysis, but I figured out how to culture and grow these cells, right? We looked at them in our microscope to compare them with the melanoma cells. We can see that they are very different, right? Dendritic cells have lamellipodia, which are flat sheets of actin, part of a cytoskeletal component, and melanoma cells have blebs. When we look at them move, they are both very fast. So dendritic cells extend and retract lamellipodia, each lamellipodial sheet about every 40 seconds, and a melanoma will extend and retract a bleb about every 20 seconds. Okay, so these, well, the dendritic cells are moving quickly, and the melanoma cells change shape quickly. And not surprisingly, given this long line of evidence, we found that dendritic cells do move in a largely amoeboid way, right? So here we have a dendritic cell where actin is fluorescently labeled. And we also fluorescently label the collagen, okay? And we see that largely the dendritic cells push their way through the collagen and find a path through, even though the collagen is very dense. But it's very easy to see that the melanoma cells don't do that, right? They clearly dig tunnels in the collagen that last, that are permanent effectively, right? All right, so then we could have guessed, in fact, that they would dig tunnels because most melanoma cells stay largely round as they migrate. But when we measure the pore size in collagen, that pore size is much, much smaller than a melanoma cell, right? So you can't move a large sphere through holes that are much smaller than the sphere, right? So something has to give here. And the obvious thing to test is that most cells, or at least the most studied form of migration, is mesenchymal migration, where cells release MMPs, they release enzymes that proteolytically degrade the collagen, right? So they chemically break apart the collagen, right? So we asked if melanoma cells were doing the same thing. And it turns out that they don't. As far as we can tell, when we inhibit proteases and when we look at local remodeling, the cells don't seem to be using MMPs to get through the collagen. This really surprised me. We're still, even as of a couple of weeks ago, doing experiments on this because I find it very surprising, right? So these cells are somehow digging tunnels without the aid of MMPs, without that enzymatic degradation. The obvious question is how? How do cells get through if they don't release MMPs? Okay. And to answer that question, we really need an image analysis framework because these images are very complicated, right? This is a single dendritic cell in collagen, and we're scanning through the 3D image, right? There's a huge infrastructure problem in general with 4D light sheet microscopy. First of all, the data sets can be large. Single cell data sets are usually smaller, but many data sets can be a terabyte or more from a single experiment. 3D data is hard to visualize, right? I've tried to ping various journal editors. I don't know how you could conceivably visualize this data in a way that doesn't break what are considered standard image manipulation rules, right? Almost all these microscopes are designed to have image enhancement prior to even visualization, right? They're at least designed to be deconvolved, but many other image analysis techniques exist, and image segmentation is still a very hard, unsolved problem, right? So many people in this field now are working in image segmentation. But it turns out that even if you do all of this, that's still not enough, right? I can segment our dendritic cell. Here I also labeled this cell with curvature. So red are areas of high curvature and blue are areas of large negative curvature. And that is informative, right? It allows us to see how this cell is poking through the collagen, but it's still not a biological discovery, right? So what do we do? How do we build an image analysis framework to understand cell form? Okay. And very few people really are analyzing this type of data. So we have a lot of white space to maneuver, but I also think that means that we have I guess, a responsibility to be simple and try and do the obvious things before we dive too deeply into esoteric applications. So I think the simplest thing we can do is relate the function of cell form to neighboring scales of organization, right? Those are the scales of organization that will most likely be able to mechanistically discover how they relate to cell form. Okay. Right, so we know that cell form is mostly created by the cytoskeleton as well as forces exerted outside of the cell, and that signaling is well known to control the cytoskeleton. Right? We also know that many of the functions of cell form are mediated by shape change and force. Right? So what that means practically is that cell form are still somewhat preliminary. They tend not to have very large dynamic range, but they do exist, right? And so we want a system that relates fluorescence biosensor activity and its dynamics to cell morphology and its dynamics. Okay. So that is still a very large project, right? So we're going to start by just considering the plasma membrane. And the plasma membrane is both a conduit and a platform for cell signaling, right? Meaning that some of the key signaling reactions in the cell take place on the plasma membrane, but also that information is transferred inside between cells, necessarily via the plasma membrane. Okay. So practically speaking, to isolate the cell surface, what we do is we generate a mesh from the cell. So here we're scanning through a 3D image of a single cell expressing KRAS V12, which is a constitutively active form of KRAS, right? And we make a mesh of the cell, which is a lot like the image segmentation problem, but it's a little different because we need to worry about subpixel resolution. Okay, and then rather than leave the intensity in image space where it's hard to access from the perspective of the mesh, we project fluorescence intensity onto the mesh, right? So here red are areas of high local KRAS V12 fluorescence, and blue are areas of low KRAS V12 fluorescence, right? And this really, this complicates the math somewhat, right? Now, rather than have an image, we have a mesh. Right? This makes it harder to measure things like spatiotemporal correlations because it's hard to measure distance on a mesh. We also can't do basic machine learning because we have no way of ordering the points on the mesh. But we now get to bring in an entirely new field. We get to bring in the field of computer graphics into computer vision. And that hasn't been done before at the cellular scale. So that really gives us it gives us a whole field of algorithms to draw from. Are there any questions? Right, so now what do we do? We still have a lot of white space. Well, the first thing I did was try and move across scales. So one key scale here is that cell morphology itself is structured. Right, so this is a melanoma cell. You see it has lots of blebs. It has many of those hemispherical balloons. And other types of cells are structured too. So I built a morphological motif dissector to find sort of stereotyped morphologies on the cell surface, right? So these are lamellipodia and blebs and filopodia. And since we know that there are many different types of these structures, I went to a summer school where we spent a whole hour just talking about the names of protrusions. We used, right, well, here's more, these are yet more, these are dendritic spines on neurons, and here I also clustered the spines by cell shape. I use machine learning to, since there are so many different types of these structures, I use machine learning to identify the structures. And rather than ask people, which is itself problematic because 3D data is hard to generate. It's hard to do supervised machine learning on 3D data because it's hard to generate training data from 3D data. Some people have used VR, but instead what I decided to do was to note that the system of naming protrusions is sort of inherently biased. It's been biased for decades. But rather than fight against that, we're going to acknowledge it and assume that it is biased and use our knowledge of human perception to constrain training data annotation. All right, so we know from very old results in the perception literature that if you give people a 3D object that looks like this, people tend to break it apart there. Likewise, if you ask someone to break apart an object of this sort, people will tend to break it there and there. They'll tend to break it along the shortest lines, right? So they called it the shortcut rule. Right, so we embedded that into our system. We broke down the cell surface into structures that people tend to identify, right? And then we had people annotate those structures, right? So I had people click on patches here, click on patches that they thought were definitely the motif of interest and were definitely not the motif of interest, right? It really mattered, we found, how you ask people to click on them. If you just ask people to click on blebs, you get an entirely different answer than if you ask people to not click on blebs. Like 40% of the surface is labeled differently, right? So we have to be very careful there. Okay. All right, so there are more details in the publication, but more recently, so we used this system for a while, and that system is obviously very biased. It's biased by the biology and biased by biologists. So we found that we wanted a way of categorizing, of breaking down signaling on the cell surface that was entirely unbiased, right? And this project is being led by Hanye, who I'm a postdoc in Gavin Stenuser's lab, who I'm mentoring, and the goal is for her to be first author on the project and for me to be last author, right? There aren't, so basically the problem we have is that cell signaling could be organized in many different ways on the cell surface. It could be uniform or punctate or patchy or polar, but the cell surface itself has widely different morphologies, right? So how do we compare signaling across morphologies, given that the morphologies are different, right? And again, we can't easily do things like measure distance, so it's hard to come to grips with how we compare signaling. Although we can't measure distance, we can transform into a spectral space, right? So if you gave me a 1D signal, the simplest way I could measure the spatial scales of organization would be to do a Fourier transform, right? Likewise, if we have spheres, the simplest thing you can do is arguably to decompose data via the spherical harmonics, okay? And it turns out there is relevant math like that for cells. It's called the Laplace-Beltrami operator. So we can decompose signaling on the surface of a cell via this Laplace-Beltrami operator. Since we're so new in this space, I don't think anyone else has done this, right? So that's what we tried. We tried generating these cellular harmonics, and they are true harmonics in the sense that if you were to turn the cells into a drum and start hitting them, that these are the frequencies they would vibrate at. Okay, right, so we can do that here. We decompose PA3 kinase activity on the surface of the cell into this Laplace-Beltrami basis, right? And this is it's completely, it's mathematically complete, but in a sense, it's almost too informative. We want to compare the spatial scales of organization from cell to cell, but that basis actually tells us too much, right? For one, it tells us, it conflates orientation and spatial scale information, right? So you can see that, say, these three modes are all polarization modes. They show you front, back asymmetry in 3 different directions, right? But I don't really care which direction the cell is pointed in. I just want to know the scale of the signaling. So what we did was to pretend that the Laplace-Beltrami basis had the same structure as the spherical harmonics. You can arrange the spherical harmonics such that each level here has a different energy. So those are symmetries in the more physicsy sense. This math is strangely similar to quantum mechanics. I think because quantum mechanics is mechanics in wave space. It's sort of spectral mechanics, right? So we average across the different levels of the spherical harmonics. And then we also convert our magnitudes into a Dirichlet energy, which just into a Dirichlet energy density, which both gets rid of the absolute magnitude of the fluctuations because they're hard to normalize from cell to cell, and the Dirichlet energy measures just how much variation there is on the cell surface, right? So now we have a spectra that more directly measures spatial frequency. So if you go from a microscopy perspective, if you wanted to test sort of the frequency properties of your microscope, you would use, or many people would use, a striped pattern. We can't stripe these cells because the stripes would run into each other, right? But we can polka dot them. So that's what we did. So here we used polka dotted cells to measure our, I guess, the frequency response of our system. So our system does do what you would expect it to, right? So here so if you just put one polka dot on the cell, we see low frequency spatial information, right? If we have more and more polka dots, we see higher and higher frequency spatial information, okay? And this allows us to do things like principal component analysis. Now, since we have an ordering of the points, we can do basic machine learning on them, okay? And we can also, very importantly, compare information across cells. Okay, and my polka dotting is not perfect. I think actually the spread here, so this is the polka dot pattern, and this is the peak spatial scale. I think the peak spatial scale is actually not as spread out as it appears. In reality, it's just that I polka dot somewhat badly. Okay. And we can use this system on real world data, not just on polka dotted cells. So here we looked at PI3 kinase signaling again, right? So PI3 kinase tends to go to the front of cells, right? And you can see that's a low spatial frequency. So you can see peaks here at low spatial frequency. And then when we inhibit PI3 kinase signaling, we see that those low spatial frequencies drop off. Okay. And yet there's one more image analysis project I want to tell you about, and then we'll go back to our melanoma cells, right? So to study dynamics, we really need to link information across time, right? And the simplest way to do that is to track the cell surface and then use that tracking to link other types of information that are on the cell surface, right? This trick has been employed for decades now for 2D image analysis, right? So there's an entire series of papers, sort of a mini literature in itself, of algorithms to track the cell surface in 2D, right? So here we're tracking, you find the edge of the cell surface, and then you also find how particular regions of the cell surface move. So say where does that part of the cell surface move from frame to frame? But unfortunately, it's not apparent how you could extend these algorithms into 3D, because 3D imaging, even state-of-the-art 3D imaging, isn't fast enough to assume that those patches on the surface don't move very far from frame to frame. And essentially every 2D algorithm assumes that the patches don't move very far from frame to frame. All right, so what I did was to step back and ask if we could think of it, rather than thinking of it as a tracking problem per se, if we could think of it as a shape correspondence problem, right? So here we can use various sorts of information to just generate some sort of local correspondence between points. So say map, the head of this person to the head of the gorilla, or the nose of the plane to the nose of another type of plane. And there are many, many shape correspondence algorithms in the computer graphics literature. So I went through the literature and tried to find one that would be best for biology. And the general approach I settled on is called functional maps. So in functional maps, rather than directly map points to points, you paint the surface of the cell via functions and then map between those functions. And then this is fast, because once you have information on the cell surface, you can do computations in the pos-Beltrami space, in spectral space, right? And I think importantly for biology, we can also paint we can paint all sorts of different functions on the cell surface, right? We could paint PI3 kinase polarization, we could paint actin intensity, or what's done here, which is shape itself, okay? And so I made a sort of prototype workflow that I was very happy with. So this is a dendritic cell. Here we're mapping 60 seconds apart. You can see that the cell surface really moves quite a bit in 60 seconds. This is blue to gold, but nonetheless, we can generate a dense tracking between those two surfaces, right? So here, green is mapped to green and pink to pink and so on. But I wanted this system to be, I don't think anyone has yet tracked surfaces in 3D, and I wanted to do it really well. So I contacted the person who developed functional maps as a postdoc, and we've been working together with his group to make the system work as well as we possibly can. So I incorporated algorithms from many different recent computer graphics papers. Really, what I think is most exciting about the framework is that we allow users to specify what conservation laws they want to use, right? So you can say that you want to preserve surface area from frame to frame, or position, or isometry, or shape, or fluorescence intensity, whatever you would like as a user. Some of those are unstable, but it still gives you freedom as a user to decide what you want to do. Right, so here we can track between cells that change their shape quite a lot from frame to frame. This is a fibroblast image that's relatively low resolution compared to some other modes, right? You see that it loses an entire branch here, it gains a branch there, and so on, but we can still track between these two shapes. Remember, light colors are mapped to light colors. And in here, I seeded points in the first frame and just let the tracking carry those points around. And I was very excited that we can even track the tips of Philopodia, which tend to wave around very quickly, because most 2D algorithms are not capable of doing that, right? But here, it just naturally falls out. Okay. And then the goal of tracking the surface is to measure motion, but also to link surface structures across time, right? So here we can track blebs across time. And we can also measure derivatives, right? So this is PI3 kinase activity at one point in time. We can use our tracking to grab signaling intensities from the next frame and from the previous frame and put them on the current surface, and that is enough to calculate derivatives. So this is a very simple derivative of PI3 kinase activity, right? And the derivative contains different information than PI3 kinase activity itself. We've also, so Robert Magnet, who's a grad student in collaborator Max Ovchennikov's lab, has also gone in and developed more derivatives. So we have a measure of deformation of the cell surface. We have vector fields, vector field measures like divergence and so on, okay? So I am hopeful that this will be a critical tool in understanding 3D signaling, right? Because derivatives are a very foundational measure of change in general. Okay. All right, so now that we have 3 different image analysis techniques to begin to interrogate this data, we can go back to our melanoma cells and ask how they are getting through, ask how they are tunneling through this dense collagen, right? So we had the hypothesis that the cells were getting through via their blebs, right? So we wanted to know how the blebs were moving the collagen. Oh, sorry. So there's collagen motion on the y-axis and cell surface motion on the x-axis. And here we're just measuring the motion of protrusive blebs. And at low motion, the cells, the blebs do what you would expect them to do, which is if they push on a collagen fiber, the collagen fiber moves. Right? That is a standard linear ballistic motion. That's just a push or a pull, right? But at higher collagen motions, or at higher cell surface motions, we find that relationship between collagen motion and cell surface motion breaks down. The bleb moves fast, whereas the collagen doesn't move any faster. And that is consistent with bleb interdigitation. So the blebs are snaking their way through the collagen, which was known before. We knew that blebs could do this. But surprisingly, we found that retracting blebs do not simply snake their way back out of the collagen, right? We lose that interdigitation regime for retracting blebs. Instead, the retracting blebs are to some extent grabbing the collagen and pulling it back to the cell surface, okay? Then the cells end up wrapped in collagen, probably in part held there by the glycocalyx. And they eat the collagen, right? So these cells are making their way, they endocytose collagen fragments, at least in part via macrophaenocytosis, right? So to measure macrophaenocytosis, you fill the area outside the cell with a sort of dye. These are large colored dextran filaments. And we see that the dextran is ingested along with the collagen, which is interpreted in the space as a sort of slurping up of the cell. So it's eating the collagen as if it were a liquid. And it is unusual that blebs can macrophaenocytose, but it has been seen especially for viruses. And viruses are not so much smaller than collagen filaments, right? So the cell is sort of eating its way through this collagen as if you might imagine someone eating their way through Jell-O. So we put it in an environment that is very soft, that it can't really gain traction on, so it's decided to eat its way through. The next question we asked is, in order for cells to migrate, they need to establish a polarization. They need to establish where their front is compared to their back, which sounds a little silly, but it's not so trivial for a cell to remember where it is going. So there is an obvious asymmetry in the cell migration, which is that it ate behind it and it is pulling at its front. So it has more adhesions to the environment and its front. Cells form adhesive structures with their environment called focal adhesions, right? These cells have more focal adhesions at their front simply because there is more collagen at their cell front, or at least in part. And adhesions are known to upregulate PI3 kinase signaling locally. We found that is what they do here. Right, so there's high PI3 kinase signaling at the cell front, right? And you can see that is consistent with what we see. There's a blebbing cell migrating in the middle there. It's a little hard to see, but there is more PI3 kinase at the front. And that PI3 kinase in turn enlarges blebs via branched actin polymerization, which we still don't understand how that works, because given what we know about blebs, that should not happen, but nonetheless it does. In general, that means, right, that the cell remembers their front-back polarization via environmental remodeling, right? So it remembers where their front is. Sorry, my kids are rather loud. Because it knows that it ate behind it, right? So there's more, it eats at the front of the cell, that upregulates adhesions at the front, which upregulates PI3 kinase, which upregulates branched actin polymerization, which makes larger blebs, which in turn eat more, right? Right, so what does that actually mean? Well, it means that cells can move without, in general, that means that cells can move without MMPs. We've known that MMPs are not, MMP inhibitors don't work well clinically for a while now, and that is probably at least one reason. It also means that cell migration should really be studied in remodelable environments and environments where the cells can alter the environment around them, because here a key element of migration, polarization, requires that we understand, requires environmental remodeling, right? And it also means that bleb size is locally regulatable via branched actin polymerization, which is not something we expected, right? So we might go back into other processes where blebbing occurs, like cytokinesis, and ask if those blebs in turn are, if bleb size is also regulated there, right? So there is a surprise. There's one more surprise that we found, which is that we can alter, when we alter bleb size, we directly alter signaling within the cell. right? So we can, when we suppress blebs, we find that it suppresses PI3 kinase polarization, right? So here we have a situation where morphology is itself regulating intracellular signaling, which is unusual in this space. Normally we think of it the other way around. We think of signaling as controlling morphology. So Andrew Weems was actually a grad student in the cell biology department at Colorado and is now a postdoc in Gowden's lab. Really followed through and found that acceptins, which are a curvature sensitive protein, localized to the base of blebs, which in turn upregulate survival signaling, right? So if you've ever done cell culture, you know that cells tend to bleb when you detach them from a surface. The thought is that they are unhappy. Here they are using morphological change. They're using the structure itself to upregulate, to control their own survival, right? Right, and there are many different ways for how morphology could regulate intracellular signaling, right? Very simply, if you change the local ratio of surface to volume, if you change the local amount of membrane for cytoplasm, we expect to alter signaling that goes through the cytoplasm, right? There could be diffusion traps or curvature sensitive proteins. I think we will find that more and more proteins are curvature sensitive. We know that different types of morphology are associated with different cytoskeletal structures that could also alter signaling. These cells change shape so quickly, we might expect induced fluid flows in the cell, and so on. Okay, so moving forward, I would like to build a computational framework that allows us to investigate how morphology could regulate intracellular signaling. That probably sounds very biologically specific, but it is actually a fairly broad framework for understanding how signaling and other subcellular structures relate to cell function, right? Relate to the next scale of organization. Okay. Right, so in summary, I've started to describe a sort of geometry for cells, talked about different types of microscopy analysis and how melanoma cells migrate in 3D. In future, I would like to use similar approaches to understand how cells regulate signaling via morphology. So what I've showed you so far is really a group effort. Right, so many people come from my PI's lab, from Gowden's and User's lab, who's known for computational image analysis, but also for cell biology. We work in the same space with two microscopy labs who have like 7 or 8 light sheet microscopes now. I've really enjoyed working with Max and his lab to bring more computer graphics into cell biology. And we've been trying to, there's a group at UCSF who's been trying to sort of make Chimera X, which is a rendering software often used for structural biology, try and make it so that people can use Chimera X to render surfaces. Right? It can do a little volume rendering, but mostly it's a surface rendering framework. Sorry, I try and leave questions in these sorts of talks. Are there any questions? Leave time for questions in these sorts of talks. Are there any questions? I know that it is probably very different than what many of you do, but we will go back to the analysis.
Thanks, Megan. That was a really great talk. We have about 10 minutes for questions, actually more. So does anyone have any initial questions? About 15 minutes.
Yeah, I thought this was really, really pretty wild. So when you talk about the cells endocytosing collagen filaments, was that on the, did that connect to polarity? So was that on the front or the back? I might have missed that. Oh,
they do it at the front.
At the front, okay.
I feel like we still don't entirely know how they endocytose the collagen. I mean, because they drag the collagen in as filaments, right? And clearly it is endocytosed as dots, right? So at some point the collagen is being broken. So the cell sort of envelops the collagen like in a sort of bleb foam. So it totally surrounds the fibers, and then it pecks at the fibers via endocytosis. So I don't know if it's the surrounding that breaks the collagen or the endocytosis. And also macrophaenocytosis is very vague. It would be good to know more about how it's being endocytosed and when it's being broken.
Do you think that process, sorry, do you think that process has anything to do with the sort of the different relationship between the leading and the retreating collagen behaviors of the cell in the collagen matrix?
Yeah, I mean, so this type of macrophaenocytosis is probably facilitated by blubbing, right? So it should be more, and that is what we see, although we didn't really measure it very carefully. It should be more endocytosed at the front of the cell, right? So that means we're mixing different processes here. We're mixing migration and macrophaenocytosis and possibly even some metabolism, because it has to digest the collagen in some way. Some people think that the cells might just be hungry. They're trying to eat collagen.
Okay.
Plenty of time. Questions? Maybe ask.
I'll ask an ignorant question. I thought it was a terrific talk. Thank you, Megan. Your really neat slide where you had the guy going like this and the mapping and function space.
I can actually see you. Let me see here. Well, you might have to describe what you mean, because I don't see your image.
The slide that you had with the mapping in function space, where you had the little sort of, they look like child toys. What that made me think of was in some mechanistic models that are dynamical systems based, as I know that you do a lot of work based on that. And I'm wondering whether there are existing computational models for any of this that could be leveraged or perhaps integrated with your work. Are there strictly computational models for cell movement or growth in it, that could be adapted?
Yeah, I mean, when I present this to physicists, they usually ask if it's physical, which is a good question, right? Because here we are trying to map the movements of imaginary monsters, so we have no reason to believe that this is real. So I think the first thing to do from a physics perspective would be to ask, would be to somehow validate it physically. I think to do that, when you preserve different when you use different conservation rules here, you have to currently upregulate surface area. And I think that that's not itself very physical, right? Because the cell can add and remove membrane. So it'd be great if we turn this more into a model that we can physically, effectively a model that we can physically test, right? By doing things like allowing the system to add and remove surface area. Yeah, that's not as complicated as dynamics, but I don't know if we're quite there yet. I tried as a grad student to see if we could understand the amoeba as a chaotic system. Remember, I had just a very simple tracking of the amoeba, and the answer was we don't have enough information. My bet which is actually an answer that math can output. My guess would be we still don't have enough information, but it's hard to say.
Neat. Thanks.
Thanks. Yeah.
How many cells can you image in a realistic amount of time, I guess? Like, how large are data sets in terms of number of cells with this technology?
I mean, I was at about 25 cells a day if everything goes well. It's not so high throughput, but things don't always go well. We can, with the morphological motif detection, we had some example analyses in there, and we can get to statistical significance with, say, 8 cells. So it's not so high throughput, right? But the goal is to make it, or a goal is to make it more high throughput. But the surfaces themselves have many points, right? Many of these meshes are 100,000 or 200,000 bases. So we have a lot of data in that sense, but it is not statistically independent, right? You have to be careful.
So Megan, great talk, by the way. I'm Sean.
Thanks.
I don't know if you can find me on the screen someplace, but just following up on, sorry.
No. Keep
I won't use my hands to gesticulate too much.
Okay.
So there was a follow-up question to Casey. I'm just curious how much of how much you could extend your capture capabilities by modeling between time points or between surfaces. So could you learn some structures that you could then predict based on either coarser timescale or coarser resolution to be able to improve the number of cells or your data capture capabilities?
I mean, we could. I mean, there are a lot of people in this space. If we go back, I guess, to there are many, or there are quite a few papers that seek to extend resolution in one direction into another direction, right? To the extent that the last time I saw a talk by the Nature Methods editor, she was complaining that there's now a family of papers called care clones that say, since most microscopy modalities have worse Z resolution, for example, than XY resolution, you could imagine using deep learning to improve your Z resolution, right? And since the cells have themselves, at least in our system, they have no preferred direction, you should have a lot of information that you could extend into Z, right? So people are already doing that. I mean, I haven't tried to do that. We did try, not me, but an sorry, I was trying to find, not me, but there we go. I guess I would classify that as an image enhancement technique. We did have an undergrad in the lab work on a deep learning based deconvolver, right? Because deconvolution is slow and the microscopists were annoyed, so they wanted something that was faster. I mean, that worked okay, but yeah, I mean, you definitely can use image enhancement. I think someone at the Allen Institute for Cell Science, I think they abandoned their light sheet and are going for image enhancement, right? But I still like the light sheets.
I guess just at least in radiology, folks struggled a lot with trying to bridge in Z space until the resolution of CT and MR slices got it so that the images are isotropic and the gap in between slices was the same as the gap in between pixels effectively horizontally. So I don't know if the same principles hold, but when they got to isotropic, the images got much better.
Okay. Yeah, I mean, so I guess the answer is yeah, that there are ways to enhance the images. On the other hand, there are also many different flavors of light chain microscopy that allow us to take different sorts of images. So we can also just experimentally acquire these images now. And we could easily morph from image to image. I didn't do that because it felt a little bit deceptive. And it's hard to tell what our actual temporal resolution is.
Any other questions? I guess I do have one. Or Casey, maybe you were going to go. Okay. So Mike, I have a bunch of questions, but maybe my main one here is about feature extraction.
Okay.
And I work a lot with 2D images, and I understand that additional third dimension adds a lot of complexity and challenges with the analysis. What we're finding in two-dimensional with two-dimensional cell images is that representation learning based on deep learning, so things like convolutional neural networks or transformers, are outperforming some of the past 2 decades worth of research in terms of forming mathematical hand-engineered features. And so I noticed that you have a lot of hand-engineered features. That's essentially the focus. And as you increase the complexity of 3D, the field really hasn't, at least like it seems, to generate these hand-engineered features is the next logical step. So these like Fourier transforms, the Laplace Beltrami things. What are your thoughts on representation learning, and how would you approach generating maybe a higher dimensional morphology feature from 3-dimensional images? Like, would that even work?
I mean, I think deep learning for meshes has lagged behind deep learning for images, just because it's you can't do a convolutional neural net on the surface of a cell. That said, there's a recent paper by our collaborators. They figured out that rather than convolve, you could use diffusion. And that is a common theme throughout graphics. It's easier to simulate diffusion on the surface of the cell than it is to measure distances, right? So can you build up, so they built up layers of diffusion where you allow the system to find effectively the best diffusion constant and learn via diffusion. And I think that's very exciting, in part because you might be able to do, and they actually have unsupervised deep learners, but also because that is essentially what the cell does, right? The cell, in some sense, uses diffusion to measure its own geometry. So I think going forward, it would be great to try and figure out if we could bridge a deep learning architecture with reality, right? Could we model, it's like trying to model a brain with a neural network, but this is a cell, right? So we may have more hope with a cell. Yeah, but I mean, I think this field in general is probably moving more towards geometric deep learning, right? That would also enable us to do deep learning on signaling pathways. But yeah,
I think these.
Yeah.
But
yeah, I mean, I think the reason why everything is hand engineered is because it's harder to do deep learning on a mesh. But the field is getting there, especially in the last 2 years.
Yeah, I think your examples of these types of models, and then you're showing about modeling the different environments and like the thing going over the cliff.
Yeah.
Like that reminds me of like models like AlphaFold or reinforcement learning that, you might have to mess with the feature space a bit to get it into that space, but it seems like maybe that's one direction.
And also keep in mind, we have no training data, right? The system, the motif detector will generate a reasonable model from one cell, which is great, because the only training data we have is the people I can coerce into labeling the cells. But that's good, I guess, moving forward, because we want everything unsupervised. I mean, I think it would be great in the future if we could find not just unsupervised static features, but unsupervised behaviors of the cell. So there's a sort of field with flies where they try and, in an unsupervised way, find typical fly behaviors from images. So could we do that with cells? And cells are arguably more complicated than flies because they move in a less stereotyped way, but nonetheless, can we find stereotyped motions of cells, right? And that's important. I recently went to an NCI visualization workshop, and we were talking about how immune cells were just, in immune therapy, we're just getting stuck in tumors, right? So there it might be useful to see if we can derive the behaviors of cells so that we can measure how, when we genetically engineer an immune cell, how that affects its actual behavior as it moves through a tumor, say.
Well, thanks again, Megan. Really interesting talk. We're at the hour, so I think we should let people drop off. I think you should be meeting with a bunch of people later on, but thanks again.
Well, thank you. Thank you for inviting me again.